What I find interesting about proteins is that they have a peculiar position in the line between chemistry and life. A protein is just a sequence of amino acids, but when folded into the correct configuration, it can move and serve vital functions. A chain of amino acids cannot do anything until it's folded, and that spontaneous function has some interesting physics and biology behind it. A good analog to protein folding is the spin glass model, where, in a spin glass, the relative spins of neighboring atoms in a magnetic alloy dictate the energy level of the spin glass. When you cool a spin glass, all of the atoms realign themselves in an attempt to take a low energy state. More atoms aligned means lower energy. However, if the spins of the atoms are distributed randomly, there is no universal agreement on which direction to align themselves. In a protein, as the protein folds, it follows this sort of spontaneous realignment, searching for the lowest energy state dictated by forces between amino acids. If a protein has many states that are equally low energy, we say that that protein is frustrated. Frustration in a protein is its ability to find a conformation that satisfies the majority of its amino acid neighbors. Here we have a figure showing the energy landscape of minimally frustrated and very frustrated proteins. In the very frustrated proteins, there are many local minima within which the protein can find a stable configuration. This makes the folding of these frustrated proteins somewhat unreliable, as there are many stable configurations, but no global minimum in which a protein can find a stable native state. What we find in biologically occurring proteins is that they tend to be minimally frustrated. This is interesting, as it goes against the idea that amino acid sequences, while protein-specific, are somewhat random. Random configurations of amino acids will produce very frustrated proteins, so we have evolutionarily selected amino acid chains that produce and reliably follow a folding funnel. The protein folding energy funnel is a specific energy landscape where, as the protein folds, subsequent conformations tend to lead the protein downwards to a global minimum energy, converging on a stable native state. Bear in mind that the protein does not follow one set path of conformations down the funnel, but its each subsequent conformation leads to other possible lower energy states that will eventually converge on the native structure. When we engineer proteins, then, we need to look for proteins that do not just take useful shapes. We need to find proteins that tend to take useful shapes.